Good. So today we will start with unit 13, 1 3. Okay. Remember in the last class we have talked about few portions of that unit 13 unit, and that's regarding the microscopy, right? And we have seen animation regarding the microscopy, including the behavior of light, as well as we saw how light microscopy works, how fluorescent microscopy work, right? So today we will focus on the second aspect of the unit number 13, and as I said earlier that you can divide unit 13 into several different types of techniques. One of such type is recombinant DNA technology, one of such type is the molecular and cellular technique, the other type is immunological techniques. So today we will talk about the recombinant DNA technology or RDT based techniques, okay? And tomorrow we'll talk about the immunological techniques and other molecular based techniques. Because recombinant DNA technology consists of most of the part of the techniques that we usually discuss in unit number 13, okay? And if you from biotechnology background or microbiology background, you are much familiar with recombinant DNA technology. So you know about the basics of cloning and why we use different types of vectors and things like that. So we already had a very fair understanding or basic understanding of this cloning and vectors and all the systems, because we talked about this in agricultural biotechnology part, right? So here we are going to talk about that in detail. So first things first, we want to talk about cloning vectors. We will talk about cloning itself. Now what is cloning in the very first place, how cloning is done, and then we will see why we have different vehicles that involved in the process of cloning in details, okay? Increasing the volume, I'm increasing the volume till maximum, not more than that is possible right now. So this is the maximum volume that is showing here at my place, okay? So here we want to talk about the cloning at the very beginning. So I'll show you an animation regarding the cloning, and once that's done then I'll start discussing about the different aspects of the cloning, talking about the different components of the cloning including the vectors. We'll see the differences, how they are made, and how to use different vector for the process of molecular cloning. Now remember, when we say cloning, we are actually talking about molecular cloning, right? So we are cloning DNA, we are cloning a specific gene, and we know why we need to clone the thing. In very simple terms, we know cloning means making many copies of one particular gene here in the molecular cloning approach. So making multiple copies from one copy, that same idea which we can achieve using PCR. So we are going to talk about both today. We are going to talk about the types of cloning, the vector properties of cloning and vector, and then we'll also talk about the types of PCR and why we use PCR in different part. But there is a fundamental difference between a molecular cloning and a PCR, totally different thing. In PCR what we do is simply take the fragment of the DNA we need to amplify and makes multiple copy of that same DNA. In cloning we do the same, but in case of cloning we are not taking any fragment of the DNA. Even if we take the target DNA, we put it inside a vector. A vector is a delivery vehicle which takes the gene of our interest and deliver it inside a host organism, and a host organism will actually allow the copy number of that particular DNA to rise inside. That is the beauty of cloning. And cloning always associated with the idea of expression, not only just making multiple copies of the gene, but also to express that gene, that means to make the proteins out of it. So that is associated with molecular cloning, which is not in case of PCR. In case of PCR it's only increasing the number of the DNA, while in cloning we are thinking about first of all the cloning to increase the number and then second thing, the same process of expression vector, using expression vector to express that target gene, okay? When we are talking about the differences between a cloning and a PCR approach to amplify the number of genes, another interesting fact I forgot to mention is the ability to produce libraries, the gene libraries. because if you're working with PCR, like you're amplifying the DNA with PCR, in that case your aim is to make as many copy of the target DNA as you can, so that you can utilize them in different processes, right? Because PCR is not unifiedly is any unique or single process. It is always tagged with other processes to find an answer to with the sequence of the DNA. While if you continue with the cloning method, cloning also help you to construct gene libraries. Gene library means either it can be the cDNA library or the genomic DNA library. In both this case, you have all the genes that you need or you may need at some point of the research can be cloned and inserted using certain vectors, and can be stored in the host cells in the petri dishes. So literally those petri dishes are just like the books, and inside the book there are specific chapters, that is the gene of interest that you can take and read. That means you can work with them. So that's another important factor of cloning. Now, as you saw in this cloning process, we need a host inside which we provide the gene of interest. But when we talk about a host, let's say it's a bacteria, it's E. coli, then in the bacterial cell they have their own chromosomal DNA. Apart from the chromosomal DNA, we use other form of the DNA as a vector. Now usually we take plasmids as a vector, a very good example of vector, but there are also other examples: Bacteriophage, cosmid, Bac, yak. These are 5 separate types of vectors that are listed. When we want to talk about these 5 separate vectors, why we use them, their advantage, disadvantage, and in details. Okay, but the thing is, when we use this vector, the vector is independent. The vector is not a part of the bacterial chromosome. It's totally separate. It's present inside the cell. So the vector can be used to insert the gene of interest. So the vector has to fulfill certain properties so that we can tag the gene of interest with the vector, and the vector can carry it inside the cell. So here we can see that the vector can be a plasmid, a bacteriophage, a cosmid, a Bac, a yak. Now why such a difference? Plasmids are about 20 kb in total length. Bacteriophage is about 30 to 50 kb of the length. Then cosmids are, let me increase the volume a little bit, it will be helpful, I believe. Okay. Cosmids can be of 35 to 50, similar like the bacteriophage, but we use cosmid for a different reason. Bac means bacteria artificial chromosomes. 75 to 300 kb inserts are possible. Yaks, 100 to 1000 kb inserts are possible, and even more than 1000 inserts are possible in other occasions. Now the question is why we use these different vectors. Now when we start talking about these different vectors, you will realize that there is no unified question or answer like which vector is the best, which is the worst, nothing like that. It is, some vectors are useful compared to the other in different situations. So depending upon the situation, depending upon the type of the genes we want to clone, we use different vectors. Like common features of a vector. The common feature that a vector should carry: it contains origin of replication. It should carry a origin of replication. Okay, origin of replication is the DNA segment recognized by a cellular DNA replication enzymes. Without replication origin, DNA cannot replicated in the cell, right? So origin of replication is a must-have feature in a vector. In any vector, there should be a region from where the replication should begin, because if it's plasmid, if you take plasmid as a vector, then obviously that plasmid should have a start point to replicate itself, because plasmid is a self-replicating molecule, right? So you need to have something to start with this replication process. So that is one thing, origin of replication is required. The second thing that you need is the selectable marker. Now the marker is antibiotic marker, which we can see here. And remember, antibiotic marker is a negative marker, right? Negative marker. We have talked about this negative marker and positive marker already in unit number 12. Now in this case it's ampicillin resistant, it can be tetracycline resistant, streptomycin resistant. So they need this marker to maintain this plasmid inside the cell, because we need to select only those cells contain the gene of interest, and without the marker it's actually not possible to find out the cell carrying the gene of interest, because the biggest problem with cloning is not the part to clone the DNA. Cloning is far easy. The difficult part is to screen that cell containing the gene of interest inside, right? So now the third important feature, multiple cloning site, also known as MCS. Multiple cloning site is DNA segment with several unique sites for restriction nucleases located next to one another, next to each other. So this multiple cloning site, they carry this many cloning vectors to like many cloning sites means, you know, obviously many different restriction enzymes can cut that place. Example, you know, BAMH1, HIND3, SAL1, EcoR1, SMA1. So these are all restriction endonucleases, and restriction endonucleases are just like scissors which will do a specific cut in the DNA, in the vector, and where it cuts in the vector is the place where the gene of interest can be tagged, the gene of interest can be added, right? So that is one thing here. Multiple cloning sites are very important for that function, for that role. So it also provides what is known as a polylinker advantage. It means the inclusion of polylinkers into the plasmid vector. Polylinker is nothing but a tandem array of restriction endonuclease sites that very short expanse of the DNA is filled with many different restriction endonuclease site. The advantage of polylinker is that they have unique sites and insert excision facilitated, and also restriction endonuclease mapping and subcloning is also made easier with this polylinker. Okay, so that's why it's known as the polylinker advantage. It's advantageous to have the polylinker part in the plasmid. Okay, now the question is why we have, you know, these features that we discussed till this point. This feature is a must have for any DNA or any other DNA fragment, whether it's plasmid or anything, to be called as a vector. They should carry all this. But there is a question like why we have different types of vector and what are their features. Now the choice of vector determined by multiple factors. One of such factors is insert size. It is one of the biggest reasons we choose different vectors, because, you know, insert means the gene of interest, the gene of interest length. Let's say the gene is small that we want to clone, then we can use any vector, because the plasmid can only carry very small fragment, less than 10 kb of the fragment, while cosmid can carry 30 to 50 kb of the fragment. Each artificial chromosome can carry till 1000 kb or more than that even. So, you know, depending upon how much gene of interest we want to clone, we use different vectors. That is depending upon the insert size. Vector size also matters, because not all the host can hold on to the large vector. It depends, like some of them are perfectly fine to hold on to the small size vector, and if you want to deliver a large size vector like YAC, East artificial chromosome, behaving like the eukaryotic chromosome, then we need to use it, we need to utilize it for the process of you know, inside the eukaryotic cell. Restriction sites matters a lot, because the very basic idea is we cut the gene of interest from the source with an enzyme. We also cut the vector multiple cloning site with the restriction enzyme. So both of these cuts should be done with the same restriction enzyme. If we alter the restriction enzyme, then it's not going to work. We need to use the same restriction enzyme to cut them, because we not always get the same restriction enzyme to do that. That's why we need to use different vectors. And last thing is cloning efficiency. Like the degree of cloning efficiency matters also, because the cloning efficiency is not fixed to all the vectors. You know, the cloning efficiency for certain host is better for the plasmid compared to the cosmid, things like that. So for example, plasmids are very famous as a vector, and, you know, literally it works, the term plasmid is synonymous with the term vector, because plasmid has excellent cloning efficiency, and also it can replicate fast. It has all the restriction sites we can put. Managing plasmids are also very easy, less expensive. But the biggest drawback of plasmid share is the insert size. The insert size should be very small, in fact the smallest. That is a problem with the plasmid. Now example of plasmid is PUC18, PUC19, PBR. These are the examples of different plasmids. So in case of PUC18 or 19, you can see they have the ampicillin resistance gene. It has a replication start site as well as the multiple cloning site, or MCS. All these sequences are present. Now plasmid's benefit is of small size, high copy number. The number of plasmid can be maintained inside the cell is huge. And also they have the visual markers for selecting the bacteria carrying the plasmid with the insert, and promoters for in vitro transcription is also possible, because as I said, the process of cloning always works keeping one idea in our mind, that is the translation of those genes into the product, and that is really, really important. That can be done with the help of the plasmid vector, keeping that in mind. So advantages: small size, origin of replication, multiple cloning site of our choice can be provided. Selectable markers are visible markers are possible. Some are expression vectors, so we can use them to transcribe into RNA and translate that RNA into respective proteins. So disadvantages, however: upper limit for clone DNA size, that is maximum 12 kb, not more than that, requires the preparation of competent host cells, and inefficient for generating genomic libraries. That is another big problem. Inefficient for generating genomic libraries. Preference for smaller clones to be transformed, and it is an expression vector. There are often limitations regarding the eukaryotic protein expression, because plasmid can carry a very small section of the DNA. So for eukaryotic genes, they are big, so it's not possible to clone that eukaryotic thing, or also to express an eukaryotic gene with plasmid. Now otherwise plasmid is the best. Otherwise there is no doubt that plasmid is best. Now from modification of the plasmid, we go with the second type of vector, that is bacteriophage lambda. Lambda phage, it's a virus, right? And the viral genome that can cause because of its particular behavior can be taken as a vector, because the behavior is they have this cos sites, right, known as the cohesive sticky ends. So cohesive sticky ends are the places which help the bacteriophage to package the DNA inside the head, and so that the phage particles can come out from the host, killing the host. Now here the protein capsule of lambda has a tight constraint, and the DNA that will fit is maximum up to 55 kb. So by the early 1970s, we know that most of these 55 kb segments are junk DNA. So we can cleave that junk DNA out. Only 30 kb of the DNA that the phage share is required for the lytic growth of the phage. So rest 19 kb are foreign DNA. Rest 19 kb are junk. So we can use that 19 kb of the length of the target DNA to be inserted, and the packaging efficiency is 78 to 100%, so near about 80% minimum packaging efficiency is present. So because the packaging is faster, means while we insert the gene of interest inside the host and we allow the host to grow, so in that case, once the number of the host gene is replicated, we make multiple copies of the host genome, and then what we can do, we can package it inside the head, inside the phage head, the lambda phage. And this packaging is also done by the lambda phage itself, because the lambda phage carries its positions like cos sites, because let's say this is the gene of interest, and to the both end we have cos site. This is a cos site here, this is another cos site here. Now they will bring those cos sites together. That will, once they join the cos sites together, that cleaves the actual gene of interest out. So that's how they can package the DNA always. Okay, so this packaging efficiency is also very good for the bacteriophage, and also it can hold on to little lengthier DNA compared to the plasmid. So that's the big deal. Now for this process, we need to modify the bacteriophage lambda. We need to eliminate the non-essential part, approximately 19 kb part. So we have the room of 20 kb of insert in it. So what we do is simply keep any other genes that are required for the lytic cycle, and also cut that 19 kb of the junk DNA out, and put the DNA of interest in that 19 kb sequence. So that's all, you know, the disarming of the agrobacterium vector we discussed. The same thing happens here as well. So types of Phage vector can be of two different: replacement vector here and insertion vector. The replacement vector says like there is the specific, this is the cos site, this is another cos site, in the middle there is a specific region known as the stuffer fragment, which will be released. So the stuffer fragment will be cleaved away, and we insert the gene of interest in between that. We can do that. It's for the replacement vector because you're replacing the stuffer fragment with the insert, while in insertion vector the idea is we are cleaving one particular location, that is here in the middle. This location we are cleaving the location and inserting the gene of interest inside that location. So it's clear insertion, while replacement vector, when we talk about this, is replacing one fragment of the DNA with the new insert. That's the difference between the two. Now, what are the advantages and disadvantages of Phage vector? Advantages: large DNA fragments can be cloned, larger than the fragments that are present in the plasmid can be cloned. Used to establish gene libraries of lower and higher organisms. You can establish these gene libraries for lower and higher organisms. Gene cloning through cosmid helps in the study of nonsense sequence in the genome of the organism. While disadvantages are the packaging enzyme fails to pack the cosmid into the phage heads if one cos site is missing. That's one big deal. If one cos site has any problem, or if one cos site is somehow missing, non-functional, then the packaging will not be done properly, mispackaging will be done, and that's one big deal. Another big problem is also associated, as it's a phage, because it's a virus, it can kill the host. So somehow if it contains the virulent factors, it can kill the host. That's another big problem here. Now cosmids. Now we've talked about plasmids and bacteriophage. Both of them are pure vectors, while cosmid is a hybrid vector. Now the name, if we divide cos and mids coming from the plasmids, cos with the cos sites. So hybrid vector. Plasmids that contains bacteriophage lambda, cos sites are known as the cosmids. So the DNA length of 33 to 48 kb cloned into restriction site, the cosmid packaged into the viral particles. Now what we do, we are using plasmids because of the simplicity of the plasmid, because of the high copy number possibility of the plasmid. That is fixed always. Apart from that, what we are also doing is we are modifying the plasmid a little bit. What kind of modification we are doing? We simply add cos sites in the plasmids. Why we are doing or adding cos sites? Because we know with cos sites the packaging efficiency will be increased much. So with the cos site we have a very high packaging efficiency. On the other hand, we use this plasmid's other features for the cloning purpose. So we get the benefit of a plasmid for its simplicity and maintenance, and we also get the benefit of a bacteriophage lambda because we have the cos sites, so higher packaging efficiency. That's why cosmid proved to be a very important vector that can clone a bigger length of the DNA, as well as better efficiency of the cloning, as well as better packaging efficiency. Okay, so cos site is the only requirement for the packaging into the plasmid in this case. Okay, although it's not the phage itself, so it cannot kill the host. Okay, so the infected cells can grow into normal colonies. That's no problem with it because it's not the entire phage. Now the fourth type of vector, bacterial artificial chromosome or BAC. Bacterial artificial chromosome or BAC, we use them because in this case, let's say bacterial F plasmid is cut with a LAG-Z gene with restriction enzymes, and once we cut it, we put the gene of interest inside. Now BACs are totally different because, you know, when we talk about the plasmid, cosmid, till this point we know the rest of the plasmid DNA is big and the gene of interest is small, while in bacterial artificial chromosome the concept is totally different, where you can see the vector size is very small and the insert they can hold up to is pretty big. And BAC once clone the entire genome. BAC can be used for cloning the entire genome of bacteria, of a prokaryotic organism. The advantages, however, here is with this vector it's possible to study larger genes, several genes at once, or entire viral genome cloning is possible. and it can live in bacterial cultures instead of requiring their endemic mammalian cell cultures. So we can put them in bacterial cultures as well as which is extremely difficult to maintain in the mammalian culture, so we can do it in the bacterial culture itself. So if you want to clone a viral genome, then we need to carry it through the mammalian cell. So mammalian cell culture and cell line to be maintained, which is not in case of bacterial artificial chromosome. We can easily maintain them in the bacterial cell, which is far easy to do and far less expensive. They are helpful in the development of vaccines, because as we're dealing with the different viral genomes, we can clone the different viral genomes, and also we can clone any particular part of the viral genomes which we want to develop the vaccine against. So that is also very beneficial to develop vaccines. And the fifth vector is yeast artificial chromosome, or YAC. Yeast artificial chromosomes are special linear DNA, linear vector that carries many different components of a eukaryotic chromosome, and those components can be telomere at the end, it can be centromere in the middle, and many genes are present surrounding it. But those genes are required for the replication process, and all these things, origin of replication, this ampicillin resistant gene as a selectable marker, all these things are there, multiple cloning site. But the place for the multiple cloning site which we can add the insert can be up to 1 MB, and that is really good, because while we are concerned about cloning the eukaryotic gene, which is very difficult with plasmid or cosmid, but with the help of yeast artificial chromosome it is possible, because yeast artificial chromosome, once inserted inside the eukaryotic cell, it can replicate itself, it can make the copy of the mRNA that can be translated into the protein. So these things are quite easy to obtain because of the nature of the yeast artificial chromosome. It carries a telomere, centromere, so everything is fine regarding the chromosomal division, cell division, and all the processes quite easy to obtain with yeast artificial chromosome. Now we all know that the purpose we use yeast artificial chromosome is to clone inside the eukaryotic cell and to clone very large fragment of the DNA till 1 MB, that should be okay. And also the features they have is acting as artificial chromosome inside the eukaryotic cell. Now, as I said, another important features of the gene of the cloning is forming libraries, and the library means all the genes that will be kept inside the host, stored in inside the different hosts. So the gene specific, like for an organism, we take all the genes, we fragmentize all the genes, clone them separately, and we make different plates with that clone. That will be termed as a gene library. So the gene libraries can be divided into two different types, genomic DNA libraries and cDNA libraries. So gene library is a collection of different DNA sequences from an organism, each of which has been cloned into a vector for the ease of purification, storage, and analysis. So it can be divided into the two. So genomic library is made from the genomic DNA, while cDNA library is made from cDNA copy of the mRNA. Genomic DNA libraries can result, when we look at the genomic DNA libraries can be prepared for every single gene that the organism have in its genome. So literally the whole chromosomal DNA. So you take the chromosomal DNA, we do the gene fragmentation with restriction endonuclease, because let's say in one genome, the total chromosomal DNA, we use 4 different restriction endonuclease to generate 5 different fragments. So from those fragments we can clone each of these fragments with the specific vector and produce the recombinant DNA, and then we do the cloning with it now and store them inside. So chromosomal DNA can be restriction fragmented, then it can be inserted to the vector, and recombinant DNA can be made, and E. coli is taken as a host where the recombinant DNA is inserted, and the process will continue. So how we do actually construct? While we produce the recombinant DNA with every single gene that organism share, then we put it inside the host separately. So let's say if that organism has 20 genes, so there will be 20 separate 20 separate vessel where we put their respective host, inside of which we have the recombinant DNA. So here only 4 genes are shown with the color coding of red, green, yellow, blue, and yellow, and you can see then we transfer that with the help of transformation inside the bacteria, which is E. coli, and then we allow them to grow. We select only those colonies which are capable of uptaking the recombinant DNA properly. That we can do with the help of the selectable marker. But once that is done, we store them so that in future whenever we need it, we can actually go back there, we can check in which plate those cells grow, those genes are growing, and we just simply take we can extract it out, and then we can do the process of sequencing to find the analysis of the gene as well. Okay, so that is not only to store them for future use, because someone may use a particular gene for the research purpose, and once the library is prepared, a researcher can go back anytime in the library to find the gene of interest and can work with it. And in this case we can preserve the library. We need to continue to preserve the library. Differences between a genomic and a cDNA library. A genomic library contains every single component of the genome. It contains the coding region as well as the non-coding region. So coding region is fine. Non-coding, promoters, introns, intergenic sequences, non-expressed genes, everything is present in a genomic library. So when we're talking about a genomic library, we're literally talking about a fragment of the chromosomal DNA that carry everything, that carry the coding, non-coding sequences, everything. And when we are talking about cDNA library, they carries only expressed genes, transcription start sites, open reading frames, and splice points. No, nothing else. So these are the things that they actually carry. That is one big difference with genomic DNA library with cDNA library. Now, how exactly we prepare cDNA library and why we need to use cDNA library? The understanding is, in genomic library we are literally breaking the chromosomal DNA into fragments and we are cloning every single fragments. While in cDNA library our idea is to utilize only the coding regions, especially in case of eukaryotes, because in prokaryotes there is no difference between cDNA and genomic library. They are the same. There is no reason to do cDNA library, to form cDNA library. While in eukaryotes, we know if we prepare the genomic DNA library, it carries it produces many things. It produces the library with all the components. So what we need to do when we want to work with only the coding regions of the DNA? So in that case we use mRNA as a template to produce the DNA, which is known as the cDNA, and then we make a copy of the cDNA, then fragment that cDNA genes and then clone them for producing or making a library. Okay, so common vectors used in library, we know plasmids, modified lambda phages, artificial cloning vectors like BAC and YACs are used, all depending upon the number of length of the inserts. For plasmids till 10 kb, modified lambda phage for 20 to 30, 40 kb, artificial cloning vectors like BAC 100 to 150 kb, yeast artificial chromosome 500 kb of the sequence, 2000 kb of the sequence are used sometimes. Okay, now how exactly we use it for the cloning? Yeah, that's quite simple as well. This is the genomic DNA, we fragment it, and let's say this is the lambda phage we take, and we know lambda phage has specific site, let's say BAMH1 site, to cleave that place, and we insert the gene of interest in this place. And we know that while the lambda phage is packaging their phage head with the genetic material, then the process is something like involving the cos sites a lot, known as the contigs, right? Now the contigs, when we talk about it's like cos sites, and between two cos sites there is a gene, let's say gene 1, between these two sites gene 2, gene 3, gene 4. So what we are doing, these cos sites are utilized to package the fragment inside the head, inside the phage head. Okay, so they always utilize the cos site to package the DNA inside the phage head, and then the phage will infect the bacteria. So we can easily find whether the phage carries the gene of interest by looking at the here plaque forming unit. Okay, and we can go back and check once the cloning is done. Now for the cDNA libraries, as I said, we are not using whole gene, we are using only mRNA transcripts, and among the mRNA transcripts, it's the processed version of mRNA transcripts that we're using, without untranslated region, no introns, tissue specific, nothing is there. So what we are simply using here is an mRNA which is mature, okay, mature mRNA. That means that mRNA already have undergone the process of splicing, polyadenylation, capping, everything is done. So we use that mRNA as a template to make a complementary DNA, single-stranded complementary DNA, and then we use that single strand of the complementary DNA as a template to make another strand, so become double-stranded complementary DNA, and then double-stranded cDNA recombined with the vector, and after the recombination is done, then we put it inside the E. coli, okay, inside the host, and they will recombinate inside the E. coli cell, okay. So that is the process. We only want exons, we only want the coding regions of the DNA to be used, okay. So importance of cDNA libraries. No cDNA library was made from prokaryotic mRNA because we don't need them in the first place. We need cDNA libraries because they are required for the eukaryotes, because eukaryotes have a lot of genetic components which are non-coding. We need to exclude all of it. We need to only select the coding sequences, and that is possible if we eliminate any other non-coding regions, keeping the coding regions. So the steps of cDNA library preparation begins with mRNA isolation and purification. After that, checking the RNA integrity, then fraction it and enrich the mRNA, then synthesize the cDNA. Treatment of the cDNA ends so that it can be ready to be cloned, and then the ligation with the vector. Then we insert it inside the host and the process. Now, among all these steps, one more important step is the isolation of mRNA and how we can do the isolation. Now, if you remember, in eukaryotes, we have mRNA, tRNA, rRNA, a mixture of all the RNAs. Now, from this diverse concentration of RNA, how can we take mRNA only? How can we isolate mRNA only? The way we can do it is utilizing the poly A tail of mRNA. So most eukaryotic mRNAs are polyadenylated at their 3' end. Okay, they are polyadenylated at the 3' end. So use that polyadenylated form or 3' end to bind with the oligo-DT matrix. So what we have is beads, and on top of beads we have multiple T-rich sequences known as oligo-DT. Now oligo-DT is the sequence where poly A should bind, because you know rRNAs, tRNAs don't have this, only mRNA have this after the maturation step. So among this mixture, only mRNA with poly A can bind with oligo-DT with the beads. And then what we do the washing step, and then we release this attached mRNA from the beads. So we only get the attached mRNA in our hand. So once we get the attached mRNA in our hand, the purification is done. Then we continue with the process, and the process starts with the simple process of taking an mRNA in our hand, and we put the multiple T, okay, hybridize with oligo-DT primer at the beginning, and then we utilize we using this RNA to produce the DNTP. So we apply DNTPs, we provide DNTPs to extend the primer. So green colored is the DNA, and red colored is the RNA, okay. So it's making the green colored part, or the DNA, single stranded DNA, we call it complementary DNA, and this DNA is prepared by an enzyme, reverse transcriptase enzyme, okay. So once the DNA is prepared using an RNA template, then that RNA template that we have earlier here, we also add poly G at the end, okay, and we then use alkali to remove the RNA. Separation of DNA RNA hybrid is done with the alkali, and then we add oligo-DC primer, because we already have added G, so we can add oligo-DC primer which will bind to the G, and then again 3' hydroxyl is free, so we add nucleotides to synthesize. So both the strands of the DNA can be synthesized. So this is a process not only hold true for the process of cDNA library, but also the same process of preparing cDNA at any circumstances. You will see the complementary DNA to be produced and utilized in cloning. You will see that to be utilized during the PCR, known as the reverse transcriptase PCR. The process is the same in all these cases. It means we are producing a DNA, double-stranded DNA, from a single-stranded mRNA template, and that's how we do it in all the situations. But now, after preparing that complementary DNA, the process will be a little different. In this case, we will modify both the ends. Actually, we are adding a linker DNA known as EcoR1 linker DNA. We add the ligase. It will ligate the linker DNA with the cDNA. Okay, so cDNA is ligated to the linkers in both the sides, and then we use EcoR1 to cleave at particular locations of the linker, because you know the problem here that originates is the cDNA that we prepared naturally does not have any kind of restriction endonuclease site. So that's why we need to use a linker. A linker is a stretch of the DNA which carries the restriction endonuclease site of our choice, which we can add to the both the terminal site of the gene. So now as we treat it with EcoR1, we know the EcoR1 cleaves after GA TTC, so it will recognize the sequence and cleaves after the G, so it will generate sticky end in both the direction using the linker, and then we ligate it with the lambda, okay, inside the lambda phage, and we can use lambda phage for the cloning of the process. Okay, so that is the idea of the cDNA library preparation using the vector. Okay, and then the lambda phage will infect the host, and if it successfully delivers the gene of interest inside, and if it successfully infects the host, then in the petri plate with the lawn bacterial culture, you will see plaque forming units. From there we can identify, right? So this is the whole process of cDNA library cloning.